Good evening, and thanks for joining us for this very special occasion, having our guest speakers, Alexis Minchaca and Georgia Porter, both PhD students working under Cantu-funded cancer researcher and award-winning cancer researcher Angelica Merlot, who's not able to join us tonight because she's not long had a little baby, so that's really exciting. And doing really well from all accounts. So I don't want to take too much of your time, but I'd love to hear what you girls have got to tell us.
Yeah, so as you said, my name's Georgia and I'm here with Alexis, and we're really excited to be here and talk to you guys because we're very grateful for all of the hard work you guys put into research. Sorry, I'm researching into fundraising. So we're both PhD students at the Children's Cancer Institute. And today, I guess we just want to run you through sort of what kind of research we do and, you know, the impact of cancer as a whole on individuals and families, and really just to thank you and show our gratitude for all of the work that you guys do. So with that, I'm going to just run through some run through some sort of statistics about cancer in Australia. And I guess this is no secret, but most people in Australia will be touched by cancer at some stage in their lives, you know, either receiving a diagnosis or their family or friends, loved ones receiving their diagnosis. You know, I know that my family has been impacted by this, and I'm sure many of the people in your group have also. So it is a really serious and important thing that we need to work on. In terms of statistics, 1 in 3 men and 1 in 4 women will be diagnosed with some form of cancer by the time they turn 75. And if you jack that age up to 85, it ends up being 1 in 2 people. So it just gets more and more severe. And in 2014, it was 30% of registered deaths were attributed to cancer. And I believe that statistic is still the same. Cancer is actually an umbrella term. And so it relates to a really wide set of heterogeneous diseases. And this is by no means an exhaustive list, but you may recognize them. And what it essentially is when malignant cells form in the tissue of which the cancer is, you know, named after. And one of the main issues that researchers face is really finding a way to target the cancer cells while not damaging the healthy cells. Each of these cancers have widely different prognoses. They have different treatment options, risk factors, and really need to be considered kind of as like different diseases, although there are things that tie them together. And so with that, Alexis's focus is brain cancer and mine is pancreatic. And so what we're going to do is sort of run you through what it means to be diagnosed with one of these cancers and then what our projects are focusing on. But first, I just want to tell you a little bit about medical research and how that has improved the prognoses and the survival rates for patients suffering from cancer. So over the past 3 decades, what we've seen is an improvement of 20%. So we've seen quite a significant increase in the survival of patients who are diagnosed with cancer. And this is really due to medical research and changes in the way that we treat patients. But unfortunately, it's not consistent across all cancer types. So one positive example is in childhood cancer. 60 years ago, cancer was almost always a death sentence for children. But today we see that it's more 8 out of 10 will survive. And we still have a long way to go in terms of reducing off-target effects and you know, and ideally to get 10 out of 10 children surviving, that's the goal. So there's still a long way to go, but that's an incredible improvement. And it's really due to advancements in medical research, collaborations between clinicians and researchers, and a push towards personalised medicine. So that's something that the Children's Cancer Institute is really kind of leading the way in. But on the other end of the spectrum, we have some cancers that have seen really, really limited improvement. And one example of this is pancreatic cancer. This is the one I work on. And what we've seen over the last few decades is that the 5-year survival increase has gone from 3.4% only to 8.7%. So really limited. And then also in brain cancer, which I'm going to let Alexis talk about because she's a specialist, but we've seen, you know, it hovers around the 20% mark. There's not been much change. So these, you know, I mean, they all need a lot of work, but in terms of these cancers, they have really devastating impacts. So this is where I'll pass over to Alexis to tell you a little bit about our group and a bit about her project. And I will let you take the screen.
Okay, yeah, I think I have the control. Thank you, Georgia, and good evening, everybody. Well, I think with what we've heard until now, it's clear that there's still a big effort needed in the field of cancer research in order to be able to effectively treat every single cancer patient. So that's why we are very thankful with all your work and support to the field. And as Georgia mentioned, we both are studying our PhD at the Children's Cancer Institute, specifically in the Cancer Therapeutics and Targets group, which is led by Dr. Angelica Merlot, who is in the picture here in this slide, and who's our recipient of the CAN2 Foundation grant. So in general, our group focuses in trying to understand the mechanisms by which cancer cells can grow and adapt to their harsh environment. Also, we want to comprehend why current treatments become ineffective and to develop new therapeutics that will allow us to improve the delivery of drugs to the tumors and hopefully getting better outcomes. So to jump into our specific projects, I would like to start by talking about stress. So if we think about stress, I'm certain that most of us have dealt with a stressful situation at least once in our life. And if we think about it, our behavior changes when we are under stress, right? So the same thing happens to cancer cells. In fact, a cancer or tumor is constantly affected by different stress signals. For example, they have to proliferate really fast, or because of the therapeutics that we are using to target them. So how they deal with this stress? What do they do when they are stressed? Well, a tumor cell in the presence of these stress signals develops this stress state that in cellular level, it's called ER stress. So the same way we are told that we need to meditate or breathe, tumor cells upon the presence of ER stress activate anti-stress mechanisms that in this case, for example, UPR is one of those. So in normal conditions, this UPR anti-stress mechanism will result in a balance between survival and cell death. However, tumor cells are able to hijack this anti-stress UPR mechanism in order to promote this survival pathway. What this means is that upon this stress, the activation of the survival UPR lets this cancer cell adjust to stress conditions. It promotes their growth. It even helps the cell to migrate to other sites in the body. And also, of course, it helps them to evade death. So how they do all these? That's exactly where our research focuses. And from the different mechanisms that they can use in order to promote these survival upon stress, we are focusing on specifically two. One of them is the transmission of this ER stress, which is a mechanism that Georgia will talk about more later. And the other mechanism that we are studying is one called the translocation of chaperones, which is what my project is based on. So what's this translocation of chaperones? What does that mean? Well, I'm not gonna go into very technical details. I just want to point out that, as I was saying, in a cancer cell, as you can see here in the picture, that has the UPR anti-stress mechanism activated, it causes an increase in the expression of some proteins called chaperones, which are represented by the colorful bubbles in this figure. And what's interesting is that it has been reported that some cancer cells, when the UPR is active, can actually translocate these proteins from the inside to the outside of the cell. And when they are sitting in the surface of this cancer cell, they actually have pro-cancer functions. And another very interesting aspect of this is that normal cells do not have chaperones at the surface. So when we look at these two characteristics, the fact that cancer cells have the chaperones in the surface and normal cells don't, and the fact that these proteins are helping the cancer to survive, we came to question if these surface proteins can work as actual drug targets in order to prevent the tumor development. So for my research project, we decided to focus on brain cancer as a model. So basically what my research is doing is trying to see if these proteins in the surface can work as anti-cancer targets in this cancer type. So the first thing that we are working on is trying to identify if actually brain cancer cells have these proteins in their surface. If they are, why they are there? What are they doing? So we want to identify the processes in which they are involved in order to promote this cancer survival. And understanding these two basic concepts will help us to actually develop new therapeutics, which in this case are represented by these blocking agents, with the aim to have these blocking agents recognizing the surface proteins in the cancer cells, which are absent from the normal cells, and actually helping us to directly target and kill the cancer cell. So why we chose brain cancer? Well, unfortunately, brain cancer is a highly deadly cancer. Around 1,750 people are diagnosed with brain cancer each year, which is around one person being diagnosed every 5 hours. And it still kills more people under 40 years than any other cancer in Australia. Sadly, this is the disease that kills more children as well. And it's so hard to treat it. The current treatment is still not able to be as effective as we would like to, that just 2 out of 10 people diagnosed with brain cancer will survive. Despite all the efforts of the last 30 years, as Georgia was mentioning before in the graph, the 5-year survival rate has increased just 1%. So that shows that there's still a lot of research needed to be done in order to understand how these tumor cells work and to find new therapeutic strategies to target these brain cancer cells. So that wraps up my PhD project, which I'm conducting under Angelica's supervision and Dr. Maria Cavallari's. And with that, I'll hand back the word to Georgia.
Oh, just first. Okay, thank you. All right, so yeah, so Alexis has taken you through a bit about the stress system and brain cancer. And before I get back into those stress systems, I'm just going to tell you a little bit about what pancreatic cancer is. Of course, it's when malignant cells form in the pancreas, which is that gland located behind the stomach, which produces digestive enzymes and insulin. There are two general subtypes. One is called adenocarcinoma, which accounts for 90% of the cases. And this is the one that I focus on. And that's the one that comes from the part of the pancreas that produces enzymes. On the other hand, we have neuroendocrine, which is responsible for about 10% of all cases, and that comes from the part that produces insulin. Pancreatic cancer is not one of the most common. It's the 10th most common cancer in both men and women. But despite this, it's actually the 5th most common cause of cancer death. And the reason for this disparity is that the 5-year survival rate is an alarming 8.7%. So what this really means is that less than 1 in 10 people are going to survive the 5 years following diagnosis. So that's obviously, very, very important that we work on this, because it's incredibly devastating for both the person being diagnosed and their families. But so why is the prognosis so poor? Well, the first, there's many reasons, one of which is that we really don't see any symptoms for this cancer until it's too late. So when the onset of symptoms does occur, we see things like appetite loss or, general malaise, which means kind of just not feeling well. And it's really hard to pinpoint, okay, you have pancreatic cancer, let's get onto it early. The second thing is that we have no screening in Australia for this. So when you look at cancers like breast or colon, we know that we have really efficient screening, which really helps us improve those survival outcomes. And so because of this, we end up finding pancreatic cancer quite late. And that means that we often have sites of metastasis when it's diagnosed. And that's when, the cancer goes and sets up shop in other organs and really just does a lot of damage. And then finally, the current treatments that we have are just ineffective. And that comes down to, we really need to better understand the biology and the mechanisms by which this cancer grows, so that we can develop therapeutics that target those cells better. And so that's sort of the pathway that we're on. And just to bring you back to those stress pathways, in pancreatic cancer, these are really significant. We know already that these survival UPR pathways are active in pancreatic cancer, and it does contribute to growth and migration and the evasion of death. But what my project sort of really focuses in on is what's called the transmission of ER stress. And I will explain this in a moment, but before I do, I just want to bring you into something known as the tumor microenvironment. And what I mean by this is often when we talk about cancer, what we think is a big mass of tumor cells. And this is true, but it doesn't really involve the complexities of what it really is, because there are many, many other cell types. And in the context of pancreatic cancer, we have stellate cells, which are named because of their star-like appearance. There are also immune cells, which are becoming more and more important in cancer research, and vascular cells and many other cell types. So what we know is that these cells can talk to each other, they can communicate, they affect each other in really significant ways. And again, in the context of pancreatic, the stellate cell and the tumor cell and their relationship is really significant, and it affects resistance to chemotherapeutics, and it affects migration and growth of the cancer. So this is really where I'm trying to focus in for my project. And interestingly, they talk about their stress together. So what we find is, and to use Alexis's example, you know, if you're hanging out with someone who's super stressed, I often find it can kind of rub off on me, and you feel that kind of energy affects you. And this happens in a different way with cancer cells. And what we do know is that they can transmit these signals to each other and stress each other out. But what we don't know is how they do that. And that's something that I really want to focus in on in the next few years of my PhD. But what we do know is that this transmission impacts proliferation and migration. So this is the beginning of my PhD. I've discovered that this transmission has a significant effect on these two things. So when we dig deeper and we can work out what the mechanism is, what we intend to do is to be able to really target in on that mechanism of transmission. And in doing so, we hope to prevent the proliferation, so the growth and the migration or metastasis of these cells, and really just, you know, stop the cancer in its tracks. So that's a really general overview of my project. And I just want to sort of give a little conclusion to everybody where, you know, brain and pancreatic cancer has still seen very limited improvements to survival, as you saw in those graphs earlier. But research is really expensive, and it takes a lot of time, and scientists spend a really long time writing grants and proposals for funding. Angelica herself, you know, it's almost a full-time job at some points, but she was lucky enough to be a recipient of the CAN2 Foundation grants for young investigators. And these are really special grants because they give early career researchers with, you know, fresh and energetic ideas a platform to really push forward in their research. And so on that note, I really want to say thank you to everyone. It's incredible everything that you guys do for, you know, us. It means that we get to do another year of research and really follow what we love and what we really want to change to these patients, but we can't do it without people like you. So from both Alexis and I, we would like to say thank you to you guys, and of course to our supervisors and funding bodies. So if there are any questions or anything, we're happy to take them.
Thank you very much for that, Alexis and Georgia. And you're completely right. The funding is a huge key for us. You know, in the fight. You know, I was reading some statistics recently that 10% of young researchers actually get funding each year, and that's devastating. You know, that's 90% of these brilliant minds are going off to focus on something else. So yeah, you're absolutely right. The funding is a huge thing. You know, we're doing our part to hopefully keep you guys working hard.
Definitely working hard.
Thank you.
Yeah, I guess we underestimate how sophisticated cancer is, and obviously various different forms of cancer. And every time I hear from one of you guys who's out there in the field doing research, it blows my mind, frankly, that, you know, we're trying to outwit something that is on a basic level something that destroys our body. And the same thing, I guess, the parallels with coronavirus at the moment, that, you know, that you just look at the amount of money that's being put into that, comparatively speaking, and the economic decisions being made around that. And I guess it's the same kind of fight.
Yeah.
Do you find it frustrating that cancer's been killing people for so long and has so little funding versus something that's just emerged in the last 6 months and, you know, billions of dollars are being thrown at it?
I'm not sure if I would say frustrated. I think the COVID situation definitely requires a bit of attention at the moment.
Yeah.
And I understand why it is going to make life a bit hard for us in the coming years, but I do believe that there's a significant body of people who care about it, and the more we can do, you know, sort of things like this even, and talk to people about why it matters, I think the more visibility, the more people will be interested in funding things. So yeah, I think frustrating in some ways and not in others.
Yeah, understandable, but maybe yeah. I guess for me it's, you know, there are the parallels that there's probably nobody that I know who's been untouched by cancer, and I guess globally everybody's been touched by coronavirus.
Wow, that's true.
Yeah.
That was a really great presentation by both of you. It was really nice and clear, so thanks for that.
Yeah.
And it's just really nice to, I guess, get an insight from the researchers about how the money that you get from their grants is sort of being spent, and you know, that just gives us as fundraisers incentive to sort of continue fundraising. So I'm really grateful that you've taken the time to share with us sort of your research. I'm a researcher myself, so I guess, you know, there's probably some specific questions that I could ask, but I guess for me, it's really exciting to see people doing some research on stress and how that sort of relates to cancer itself. And I guess like one of the questions that I had was like, if you're, in terms of like you spoke about how if your cells are under stress, the chaperones head to the cell surface, and you can use those as sort of like, you know, targets for drug therapy potentially. And like that you'd indicated that that's, you know, in relation to brain cancer. Like, do you know what other tumor types that similar sort of response happens to, and is it going to be specific, or might it end up being quite widespread for different types of cancer?
Yeah, it actually happens in different types of cancer. It has been reported already in breast, prostate, colon cancer as well. There has been a lot of research of colon cancer as well. And what they found is that cancer and cell type specific. So it can also vary like from different cancer types and different cells. That's why it would be necessary to study like different cancer types. So yeah, it has been found that this response happens in many different cancer types. That's why it's an attractive option.
Yeah. And have you got some potential drug targets or compounds at the moment, or?
There's one group. I don't remember if it's for breast or colon cancer that has been developing some therapies, and I think they're in clinical trials with one of the molecules trying to target in the surface. Yeah.
Okay.
So there's potential.
Yeah, that's interesting. Thanks.
I guess that's the golden egg, is trying to find the common thing between a variety of different targets at the same time, like talking about how cells communicate in pancreatic cancer. If you could figure out how they are communicating, I imagine that is something you could apply to other types of cancer as well. We hope.
Yeah, definitely. I think the research in pancreatic cancer is, for this, the transmission part is fairly limited. There's been a lot, again, breast and colon and these cancers, which do typically get quite a lot of funding. They've done a bit of transmission, but yeah, like you say, if there's a similar mechanism across the cancer types where you can sort of target that transmission, then it has the capacity to be widely used, potentially, hopefully.
Absolutely. I mean, that'd be fantastic if there was something that you were researching on specifically for pancreatic that could then be applied to brain, breast, anything, you know, that'd be an amazing, we'd love to have a silver bullet. it's the dream. But thank you for the work you're doing.
Thank you so much, Georgia and Alexis, for your contribution, and we really, really appreciate it.
Yeah, thank you so much. Really appreciate the
for-
time that you've taken.
Thank you for inviting us.
Yeah.
It was really nice.
Really
Pleasure.
fun.
Absolute pleasure. So thank you very much for the work you're doing. We're going to keep doing what we're doing, raising the money to keep you guys working. So that'll work together nicely.
Yeah, makes the 4:30 a.m. alarm on a Saturday
Oh.
much much easier to take.
There's not much that could get me up at that time, but I'm very grateful for you.
Come join us.
Yeah.
Absolutely.
Thanks, guys.
Thank you so much.
Thanks very much.
Thank you.
Thank you.
Bye.